Hello and welcome again to the microbial world. On this episode, we will be learning about gram-positive and gram-negative bacteria, the structure, examples of each. Kindly subscribe, share, like and send in your comments. Introduction to bacterial cell wall structure and classification. 1.1 Overview and importance of the gram reaction. Bacteria are universally classified into two major groups, gram-positive and gram-negative, based on their distinct cell wall architecture and composition. Their differential response to the gram staining technique, developed by Hans Christian Gram in 1884. This classification is critical in microbiology because it predicts several biological and clinical characteristics, sensitivity or resistance to antibiotics, particularly the beta-lactam drugs like penicillin, pathogenesis and toxin production, whether the bacteria produce an endotoxin lipopolysaccharide or primarily exotoxins, metabolic behavior and growth requirements. 1 point to peptidoglycan, the cell wall, which provides structure, shape, and protection, is primarily composed of peptidoglycan murine. This large polymer is a lattice of repeating sugar units NAG and NOM cross-linked by short peptides. The thickness and accessibility of this layer are what determines the gram reaction, the gram staining process. The gram stain is a differential stain where the thickness of the peptidoglycan determines the final color retained by the cell. Step reagent use function mechanism, key observation. 1. Primary stain crystal violet purple dye stains all cells purple. Crystal violet enters the cytoplasm of all bacteria. 2. Lugol's iodine grams iodine binds crystal violet to peptidoglycan. Forms a large, insoluble crystal violet iodine complex inside the cells. 3. Decolorizer alcohol, acetone, the critical step, selectively removes crystal violet iodine complex. Gram-positive cells trap the complex. Gram-negative cells lose the complex. 4. Counterstain saffron in red, pink dye stains decolorized cells pink. Colors the gram-negative cells that were stripped of the purple stain. Gram-positive retain the primary stain, appearing purple or violet. Gram-negative lose the primary stain and take the counter stain, appearing pink or red. Cell wall structure and composition. The outcome of the stain is entirely dependent on the physical barrier presented by the cell wall. 3.1 Gram-positive cell wall structure. Key feature: a remarkably thick layer of peptidoglycan 20 to 80 nanometers, which is highly cross-linked and multi-layered. Here is a typical structure of a gram-positive bacterial cell wall. Properly study this structure. This thick peptidoglycan layer is situated immediately external to the plasmacytoplasmic membrane. The peptidoglycan matrix contains teichoic acids and lipoteichoic acids. These are polymers of glycerol or ribitol phosphate that extend through the peptidoglycan and aid in cell wall maintenance, adhesion, and ion regulation. When alcohol is applied, the thick peptidoglycan layer becomes severely dehydrated and shrinks, closing the pores and effectively trapping the large crystal violet iodine complexes inside the cytoplasm, making the cell resistant to decolorization. Gram-negative cell wall structure. Key feature: characterized by the presence of an outer membrane. The peptidoglycan layer is extremely thin to 7 nanometers and lies within the periplasmic space, the area between the inner and outer membranes. Here is the structure and component of gram-negative bacterial cell wall. Periplasm. The space between the inner plasma membrane and the outer membrane is the periplasmic space or periplasm, which is large and contains key degradative enzymes. Outer membrane composition: porin proteins, selective channels that regulate the entry of molecules. Lipopolysaccharide, located on the outer leaflet of the outer membrane. Functional implications: lipopolysaccharide acts as an endotoxin. The lipid, a component of lipopolysaccharide, is released upon cell lysis, causing fever and potentially fatal septic shock. Outer membrane acts as a barrier. It provides resistance to detergents, bile salts, and many antibiotics. Sensitivity: the thin peptidoglycan makes them more sensitive to mechanical damage once the outer membrane is compromised. 4. Comparative analysis and identification. 4.1 Summary of structural differences. Feature: gram-positive, gram-negative. Peptidoglycan thick, thin. Outer membrane. Outer membrane absent, present. Teichoic acids present, absent. Lipopolysaccharide endotoxin absent, present. Periplasmic space small, large. Stain color purple, pink, red. laboratory identification and key tests. While gram staining is the first step, further tests are needed to differentiate genera. Test gram-positive response, gram-negative response significance. Catalase test Staphylococcus positive, Streptococcus negative variable. Differentiates genera within gram-positive cocci. Oxidase test usually negative, many positive. For example, Pseudomonas identifies bacteria containing the cytochrome oxidase enzyme. McConkey agar poor or no growth, good growth. Lactose fermentation is visible, pink colonies selects for gram-negative enteric bacteria. Blood agar good growth. Hemolysis patterns, for example, beta hemolysis, are key good growth. Used to assess growth requirements. Used to determine hemolytic capability. Examples of gram-positive bacteria cocci, Staphylococcus aureus dollar, text MRSA dollar, Streptococcus pyogenes strep throat, Enterococcus faecalis. Bacilli rods, Bacillus anthracis anthrax, Clostridium tetani tetanus, Listeria mo and OCYTOGENES. For examples of gram-negative bacteria, rod-shaped, Escherichia coli, Salmonella enterica. Pseudomonas aeruginosa opportunistic pathogen, cocci.